Hello and welcome to Effortless Mastery. Today we'll be learning about Oogenesis. We're still on our embryology playlist. If you are yet to watch any of our former videos, please ensure the link to the videos will be in the description. Ensure you go back, look at them, watch through our videos. They will help you a lot. So Oogenesis, what is Oogenesis? Oogenesis is just the development. Genesis means development or formation. Development and formation of the female sex gamete. Development and formation of the female sex gamete is termed oogenesis. Now, it is important to know that every sex gamete, whether it's male or female, that is whether it's sperm or egg, is gotten from one source, and that one source is called primordial germ cells. So everything we have was gotten from primordial germ cells. All right, so where are primordial germ cells themselves gotten from? PGCs are gotten from epiblasts. So during development, intrauterine development, around the second week, cells of the epiblast begin to migrate, and those cells are called primordial germ cells. These cells that migrate will move, and as they move down, these primordial germ cells, they move down face into the wall of the yolk sac. We'll get to learn more about that later. And from there, they migrate to the gonads. And if they got to the gonad called testes, of course, the name will be called spermatogonium. So based on where it's going to, males have testes as their gonads. Females have ovary as their gonads. So if this is a male, when primordial germ cells get to the gonad, which is the testes, they will differentiate from primordial germ cells to spermatogonium. But if it is females, when the same primordial germ cells from the surface of the epiblast gets to the ovary, its differentiation will be into something we call oogonium for singular, oogonia for plural. Now, the process by which oogonia differentiate to form the matured ovum is called oogenesis. The process by which oogonia differentiate to form the matured ovum is called oogenesis, which is what we are going to learn today. But for the sake of clear understanding, I'm not talking about it, I'll be talking about it in bits. So I've broken them down into oocyte development and follicular development. Oocyte development and follicular development. Now, the lovely thing here is, usually during development, the developing germ cell requires support. Listen very carefully. The developing germ cells require support. Now, for males, the support is gotten from Sertolic cells. Whereas for females, the support is gotten from follicular cells. Now, where are the sources of follicular cells? In male, it is a cell on its own. But for females, follicular cells are actually gotten from. So during follicular development, as the follicle is developing, follicular cells are actually gotten from cells we call epiblast cell, the surface of the ovary. So the epithelium that would have lined the surface of the ovary is where I get my follicular cells from. So they help in support. So when we're going to learn about oogenesis today. Our focus will not just be on the egg, but on the development outside. So that's why it's broken down into oocyte development and follicular development. Now, another way I can break down oogenesis will now be into before birth and after birth. It is important to know that oogenesis begins intrauterine. In our introductory video on embryology, I mentioned what intrauterine means, within the uterus. So when the child is not even born, that's when oogenesis begins. But when we go to our spermatogenesis video, you would observe that spermatogenesis begins at puberty for males. Okay, so now when we come back here, in oogenesis, what are the key things to take? So like I said, I can learn this development by looking at just the oocyte and then looking at the entire follicle. At the same time, I can learn this development by looking at before life, intrauterine, and after birth. That's when you've brought out the child from the uterus, extrauterine. So let's start with oocyte development. Now, development generally, spermatogenesis, oogenesis, they follow a particular sequence of stages, which is always, that's for the exact primordial germ cell. For me to move from oogonia to matured ovum, I follow 3 stages. Number 1 is called mitosis. Number 2 is called growth. And then number 3 is called meiosis. Now, what is mitosis? Mitosis is a type of cell division. We have a video on this in our playlist. I'll add it to the description of this video. Now, mitosis is when a particular parent cell divides to form two daughter cells that are identical. So they will retain the diploid number of chromosomes. So from oogonia, I will divide into many oogonia. So from one oogonium, you have many oogonia. So you'll be seeing them. Now, gradually, each of these oogonia will now grow in size to become something we call the primary oocytes. Though, of course, during development, the lovely thing about this is that as one oogonia divides into many oogonias, they come into clusters. And that cluster is now surrounded by follicular cells that will help them. That will be like a source of support for them. Now, from here, please observe, the only difference between here and here is just growth in size. And it's called primary oocyte. Now, the primary oocyte itself differentiates into two: secondary oocyte and first polar body by a process called meiotic division. Now, remember I said generally oocyte development is 3 stages. Number 1, mitosis, where one oogonium differentiates or divides to give us multiple oogonias. Now, growth in size now picks each of the oogonia to now change them to primary oocytes. Now, this oogonia will now begin meiotic division. But the thing about females is that once they begin meiotic division, all of this happening intrauterine. Once they begin meiotic division, they are now arrested. So they will start dividing meiotically, that is meiotic division. Now they are now arrested at a particular stage called diplotin stage of prophase 1. Remember, meiosis have 2 divisions: meiosis 1 and meiosis 2. So during meiosis 1, we have prophase, metaphase, anaphase, and telophase 1. So in prophase, we have some stages. We have leptotin, zygotin, pachytin, diplotin, and dikinesis. For more understanding, you can go refer back to our video. But for now, it is important to note that primary oocytes, as they begin meiotic division, are arrested at diplotin stage. And the factor that is responsible for this arresting is called oocyte maturation inhibitor. Oocyte maturation inhibitor. So it inhibits oocyte development during the while it is yet in the womb. Now, after this inhibition, what happens to this cell? It stays in the womb. And then, delivery now happens. So everything at birth, the only thing we have that has to do with development is primary oocytes. Why? By then, all of the oogonia would have done the same thing and changed to primary oocytes. And all of these primary oocytes will be arrested away diplotin stage. All right. Now, this will stay arrested up until puberty. So that means when the child is born, the neonate will be having only primary oocytes. But while the child grows and gets to the age of puberty, which of course differs in different people, we're even told, study shows that we're even seeing females at 9 see their first menstrual period, which indicates puberty, menarche. So that menarche happens, and then, so that is it. So once they start puberty, every single month, 15 to 20 people are caused to grow, and these people that are growing are selected to grow. These oocytes that are coming out, all of these oocytes, look at them. They will now move from where they were arrested, they will resume maturation and complete the first meiotic division to give us secondary oocytes and polar body. Please, what's the difference? When I divide, the ideal thing is after division, you should be having 2 cells that are equal in size. But here, one of the cells carries most of the cytoplasm. The other one carries only a small part of the cytoplasm. So the one that keeps most of the cytoplasm is called secondary oocytes. The person that carries only little of the cytoplasm is called polar body. So please, this is that for meiosis. But I go on from here. Now, after the secondary oocyte has been formed, from the secondary oocyte, second meiotic division would occur. Please observe mitosis, growth, first meiotic. Now I get to second meiotic division. The secondary oocyte will begin second meiotic division. But when it gets to metaphase 2, it is arrested. So I will begin my division, but I am arrested at what? Metaphase 2. Once it is arrested at metaphase 2, the female stays that way, the egg stays that way, and this is where the egg is ovulated. This is where the ovulation occurs. So the egg is ovulated and then it stays that way. Now, by the time it stays that way, it only resumes if fertilization occurs. Please take note of that word, if. So if there is no fertilization, that egg will not complete maturation. So that means there are a lot of females that are even in their reproductive ages. There are some females that might be up to 20 to 25, but have never been pregnant before. Those females have never had a complete oogenesis. Why? Their pathway is always arrested at this point. That after they ovulate, there was no sperm to fertilize. So if fertilization actually now happens, okay, we now say that our secondary oocyte divides completely and gives us the matured ovum, which has most of the cytoplasm, and then a second polar body, which is like the other polar body, having just small rim of cytoplasm. And that concludes oocyte development. Now we now come to follicular development. So I know what is happening to the primordial germ cell. Okay, how does the oogonia become matured ovum? This is what we observe. Mitosis, growth. First meiotic division that is arrested at the protein, and then completed at puberty. And then second meiotic division now begins, arrested at metaphase, only completed if there is fertilization. And then we have matured ovum. But at the same time, these eggs don't develop alone. They need the support of the people around them. And those people are called follicular cells. And these follicular cells are derived from the surface of the ovary. The surface of the ovary. So the ovary is an organ that should have cortex, outer cortex, inner medulla. So that means on that cortical outer part, I have an epithelium lining them. And that epithelium, those cells, they're actually simple squamous epithelium, that the cells that will come to surround the oogonia while they are developing. And as they surround them, the primary oocyte plus that follicular cell has a name, and that name is called primordial follicle. So whenever you hear us mention the word follicle, what we are talking about is a developing oocyte and the follicular cells, a developing oocyte and the surrounding follicular cells, or more like the supporting cells. So, all right, we now go ahead. So during development, the follicular development, that development of oocyte and the people outside, is actually divided into 3 stages. The 3 stages, number 1 is called pre-antral stage, number 2, antral stage, and number 3 is called pre-ovulatory stage. So let's observe them: pre-antral, antral, and pre-ovulatory. Now, what's the difference? Pre-antral stage is the stage whereby I had a primordial follicle, which was just a primary oocyte and a follicular cell, changing to become primary follicle, which is now primary oocyte, but this time around with granulosa cells surrounding him. So this initial guy I had outside, that's why I used black. The follicular cell will change from simple squamous to cuboidal. So of course, you know, squamous means flattened. Cuboidal means a cube pattern, same height as width. So of course, what happens to the cell? So I'm not just looking at the inner structure. I'm now looking at the supporting cells outside. The supporting cells outside, which were initially flattened, now change from flat to cuboid-like. And that's not the only thing that happens. From one layer, they now become many layers. So this is what we say, that in pre-antral stage, primordial follicles, which are stimulated to grow, of course, please, this follicular development happens every monthly cycle. So every single month, about 15 to 20 are selected to grow. And as they are selected to grow, from that people, you realize that, what's happening? From the 15 to 20 primordial follicles that are selected to grow, the follicular cells begin to proliferate. Proliferate means multiply by mitotic division. So it will just divide and form many layers. At the same time, the shape is changing from squamous, which was flattened, to what? Cuboidal, which is cube-like. All right, now that is all about the first stage. Now in the second stage, which is called antral stage, in antral stage, as I keep dividing, as cells keep dividing, small spaces appear between them. So I keep dividing, I leave a space. I keep dividing, I leave a space. I keep dividing, I leave a space. Those spaces add up to form a cavity, and that cavity is called the antrum. So those small space, that addition, there's an embryology, it's an English term, but that's the term we use for embryology. We say it's coalesce, Coalesce. All right, so they coalesce to form the antrum. Now, beyond that, the oocytes, together with the granulosa cells, produce a particular glycoprotein coat that protects the primary oocytes, and that coat is called zona pellucida. That coat that protects the primary oocytes, produced both by the primary oocytes and the granulosa cell, is called zona pellucida. It is actually a glycoprotein coat. Now, beyond this, from the stroma, we also have thicker follicular. Thicker follicular cells are actually gotten from the stroma. So when you go into the ovary has an outer cortex lined by epithelium and then an inner medulla. What is actually forming those inner medulla are stromal cells, or in this case, we can call them connective tissue cells. So it's just the connective tissue that lines the ovary. So within the ovary, so I have this, so it's just the connective tissue that will now come and surround. And that connective tissue is what becomes thicker follicular, which is just thicker internal and thicker external. Internal is really concerned with nutrition and helps, together with granulosa cells, they will produce estrogen. He starts the production, whereas granulosa cell is the person that will complete the production. Then for the other guy, which is external is just really for anchorage, to anchor the oocyte. So mainly support, to just support and anchor him. All right, so that is that. So that's thicker follicular. From there, we move from antral follicle, which is vesicular follicle, to the Gravian follicle. And this stage where antral becomes Gravian is called pre-ovulatory stage. Now, what are the key things to note in this stage? Pre-ovulatory means I'm trying to prepare this guy for ovulation. What's ovulation? Expulsion of secondary oocytes from the developing follicle into the ampulla where it is being fertilized, ampulla of fallopian tube. All right, so you now observe that from here, one of the things that happens is that the antrum becomes bigger in size because more fluid seeps in and it pushes my oocyte towards one end of the follicle. The antrum becomes bigger, pushing my oocyte towards one end. Beyond that, another thing that is worthy of note is that your primary oocyte becomes a secondary oocyte. So that is, I am completing meiosis 1 at this point. Now, beyond that, the granulosa cells that surrounds the secondary oocyte are called cumulus ophorus. All right, so I think that is just that. Every other thing stays as we know. So once ovulation occurs and I expel this secondary oocyte, this is its outside, accompanied by the zona pellucida, and these cumulus ophorus cells that initially surrounded it here, when they come out here, we call them corona radiata, C-O-R-O-N-A, radiata, R-A-D-I-A-T-A. So corona radiata. Now, what is left is now called corpus luteum. And corpus luteum, what changes what is left of corpus luteum is called luteinizing hormone. So under the influence of luteinizing hormone, whatever is left, we have a lot of deposit of lipids, and so we just call it luteinization, and it's now called corpus luteum. And the name, and it produces progesterone majorly. Though we are told they produce small fragments of estrogen, but majorly progesterone. And this progesterone helps in preparing the uterus for implantation. Remember, God was wonderful in his creation, that he made sure that if I am ovulating an egg to be fertilized, I need to also prepare where he has to go and implant. So the lovely thing is that at ovulation, while I expel an egg for the sperm to fertilize and complete oogenesis, at the same time, what is left, which becomes corpus luteum, produces progesterone that goes back to go and prepare our uterus for implantation. We'll talk a lot about this when we talk about menstrual cycle, which would be one of our next videos when we talk about menstrual cycle in details. All right, beyond this, the key things to now take note of in the end, in summary, the key things to take note of in oogenesis is that oogenesis is just development of development, differentiation, maturation of oogonium to matured ovum, occurring where? In the ovary. Now, when does it begin? Intrauterine life. Some notable dates I don't want you to miss or forget about oogenesis is the 5th week. This is when the primordial germs will arrive at the ovary and become oogonia. Following this time, division begins. Oogonia will start doing mitosis. And then at the 3rd month, at the end of the 3rd month, the oogonias are now in clusters surrounded by follicular cells. Remember where we get it from? Yes, surface epithelium of the ovary. Now, at 5th month, this is when we have the highest peak of these primary oocytes. So I keep dividing and dividing till I get to an higher peak. So I start from 1, go to the highest at 5th month, which will be around 7 million. And after then, it start dropping. So you realize that as a woman ages, she drops in her ability to actually be more fertile. So as she ages, she drops. So that is following 5th month, which was 7 million, they just start to be destroyed. So they become atretic following that 6 months. And even at birth, from 7 million, we have around 600 to 800,000 primary oocytes. Of course, the primary oocyte together with the follicular cell is called what? Primordial follicle. At puberty, following puberty, every single month, 15 to 20 follicles, primordial follicles, are stimulated to grow. So they will go through all these series of follicular development, which we have learned already, and they go through all of them. And ultimately, only one out of those 15 to 20 ends up becoming a Gravian follicle, and then only one egg is being released. So this keeps happening month after month. And then you realize some part of development is before birth, others after birth. So this brings us to the end of the topic of oogenesis. I am certain you understand. But if you have any form of concerns, any form of questions, please do well to mention it in the comment section. If the video helped you in any way, mention it in the comment section. If you have any suggestion of a video you want us to make, please also do that in the comment section. Beyond that, please subscribe to this channel. Also like this video and share it so I can get to as many people as possible.